Well, good afternoon, everybody. Good evening or good morning, depending upon where you are around the world. Thanks for joining us. We're excited. This is our second webinar that's covering our encapsulation technology to help dramatically improve the genetic performance of strains and biologicals pertaining to agriculture. My name is Brennan Duty. I'm a director of business development working in our agricultural practice. I come to Ginkgo after a few years at both Cargill and Land O'Lakes, where I worked in biofermentation at Cargill, doing in-licensing of technology. And at Land O'Lakes, I helped develop several businesses outside of the United States, but up and down the Americas in crop nutrition, crop inputs, as well as seed genetics. I had a startup once upon a time focusing on enzymatic approaches to controlling pests. So for those startups out there, I understand where you're coming from and can certainly understand how to work with you. And before doing business school at Cornell, where Angela went as well, I worked in Latin America for several years working in the offset market. So it's a pleasure to be here with you, and I'll kick it over to Angela.
Hi, everybody. Angela Aloni, and like Brennan said, you'll see some similarities in our background. I've been at Ginkgo for a little over a year. We've been building up this great agricultural practice and leading up the business development team here. Prior to that, I was at Calyx, a small biotech company in Minneapolis, and at Land O'Lakes before that, also in business development and strategy across the groups of Land O'Lakes, which are Purina Large Animal Nutrition, the dairy business, and Winfield Crop Uniteds, and then Truterra Ag Sustainability. Then I was at Nestle for a decade before that. I also did Cornell Business School like Brennan did, and as I mentioned before, MIT undergrad. And we're very excited to be talking to you today about our great capabilities. I'll send it over to Andy.
Hey, my name is Andy. I'm located in Switzerland, so it's almost bedtime for me. I'm the senior foundry lead of the encapsulation and screening foundry at Ginkgo. Prior to that, I was heading the overall activities of a startup company, FGen, which was acquired last year by Ginkgo. I'm the scientist on the call, so my background is in biotechnology, industrial biotech, and I have done that for the last 20 years. And I'm happy to tell you today a little bit more later about our technology platform that we have commercialized.
Great, thanks, Andy. So we've got a pretty packed agenda today. I'm trying to fit a lot of information in just 30 minutes. But what we're going to cover are the challenges that growers are facing today and how innovators like yourselves in our audience are rising to the occasion to address those challenges. We'll talk about Ginkgo and our platform and how we built it out over the past year or 2 years, and how we can support your R&D efforts to complement your existing teams and capabilities. And then we're going to do a deep dive into our unique technology that Andy mentioned, and we'll talk about from a scientific perspective, which is for rapid strain optimization, and go through a few case studies that I think will help you understand how it can be applied to your product development cycle. And then we'll close with an overview about how we partner, and if there's a little bit of time, we'll do some Q&A. So let's get started. So as everyone on this call is aware, that growers are under a lot of pressure, and they're under a lot of pressure to grow more food, but on the same amount of land. So adding to that with increasingly erratic weather, a changing climate, changing regulatory environment, and there's just a lot that farmers have to endure and overcome in order to be able to meet the challenge that we are charging them with, which is, again, producing more food on the same amount of land. So really the picture that emerges is clear. They need you, the technology innovators, to help serve them and deliver new products that meet these challenges. Now, many of you are already familiar or actively developing biological solutions to address these challenges. And biologicals, what we mean by that at Ginkgo, we're talking about live microbial products or products that can be made by microbes. So this means proteins, RNA, pheromones, plant growth regulators, to name a few. And these molecules and biologicals have long been a part of the solution to these challenges. And while they have a lot of potential, these growers expect biologicals to be potent, consistent, and stable on the shelf. But I think anybody who's practiced within this realm for a while has noticed or has heard that developing high-performing, consistent biological products has been difficult. When you're looking at cost of use and you're looking at efficacy, sometimes they just haven't been up to task. And our aim at Ginkgo is to change that paradigm. And we're aiming to change that paradigm through the tools of synthetic biology and minimize the risks in the innovation process so that we can quickly deliver products that meet the expectations of growers, at the same time while staying in tune to the demands of regulatory bodies as well. So today we're going to focus our proprietary technology to dramatically improve the performance of biologicals and do so quickly and in a way that doesn't trigger regulatory impediments. And this is so quick that in fact we can help deliver a product to you for testing in field in certainly within under a year and typically with under 9 months. So before we dive deeper into that technology, I want to talk about a little bit on our ag biologicals R&D platform. So we support product development from discovery all the way through scale up. And we support innovators who are doing product development cycle from better starting points for new products, as well as services that help improve and optimize product performance and services that also deliver more consistent and stable products. We're doing this through several things, specifically acquisitions that we made for the past 18 months, including Bayer's Biologicals R&D division. So the excellent team that deliver products such as Serenade, Poncho 2.0, as well as Zymergen and Join Bio that's been known for its nitrogen fixation program. So our proprietary stain collection and computational pipeline, starting there, it serves innovators who are seeking starting points for new products. And then for innovators who want to build on the performance of wild-type biology quickly, so perhaps many of you who are in the audience today, we have proprietary screening technology that has the throughput to match the number of variants that mutagenesis generates. And that's what we're talking about today. Then when we get to metagenomic databases, it spans more than 5 billion genes, and we've characterized countless protein and pathway designs. And we've done that, of course, not only from our work within agriculture, but across the life sciences with which Ginkgo does. And this data enables us to approach strain engineering intelligently and quickly. So we curate the protein and pathway designs that we think are most likely to deliver the characteristics. And these approaches serve innovators who are wishing to employ both cisgenic and transgenic engineering to supercharge the biological products. On the discovery and optimization services, we're just half. The other half is really on fermentation and formulation. And we have a process development team that sits at our facility in West Sacramento, and they can iterate on formulation and fermentation processes across a variety of product concepts and test the performance in growth chambers, greenhouses, and microplots right next to our facility. So truly everything from a starting point to all the way to microplot trials we can do. And at that point, we are ready to hand off an optimized product, whether that's on the genetic side or the fermentation or the formulation to you, our customers. So broadly speaking, our aim is to partner with you to help get higher performing, consistent products to the market faster. So now that we've given you an overview about how we're working with this, with our platform, we want to zoom in on how we're optimizing strains rapidly for our Generation 2 type products. Specifically, we want to talk to you about our encapsulation and screening technology, aka NCAPS. And I'm delighted to share with you that this technology has underpinned or is underpinning our collaboration with Syngenta, which was announced almost a month or so ago. And I'm also excited to share with you that just recently AgBiome, who's a leader in the development of biologicals, is also trusting this technology to help optimize some of their strains. So we're really excited that we're going to get to partner with 2 marquee companies in this space and using this technology. And with that, I'm going to pass it over to Andy, who's going to introduce you to the technology and how we pair it with massive strain libraries to help drive rapid strain optimization. Over to you, Andy.
Thank you, Bren. So what is NCAPS exactly and how does it work? So NCAPS is a screening platform that can screen millions of strains at once. So the few you are familiar with biological R&D might think, yes, I can do that with fats for quite some time. But let's say your biological product is a live microbe that secretes some active ingredient, such as a fungicide. Or maybe your biological product is a small molecule and you need to hyper-producing strain to bring your cost of goods down. FACS will not work for secreted products, which is the main difference between our NCAPS platform and previous technologies. With NCAPS, we encapsulate each strain variety in its own nanoliter reactor. On this picture, you can see these light green bubbles, which are empty ones, and the occupied ones are the ones with the red dot, in its preferred growth medium. And we link the desired phenotype that you are looking for to a fluorescent signal. So depending even on the activity of interest, An increased signal may correspond to a gain or a loss of fluorescence. So inside these nanoliter reactors, the cell proliferates and secretes its product, be that a small molecule, an enzyme, a protein, or what else. As the nanoliter reactors pass through the sorting machine, those with the greatest change in fluorescence are sorted out. The strains can be isolated from the nanoliter reactors, and the desired activity can be validated in a secondary assay, which is typically a suspension culture. So what's special about this? There are a few things I want to highlight. We are in an era where we have affordable and fast ways to generate a lot of genetic diversity from a starting strain. So classical mutagenesis is a very commonly known technique if you want to be in the non-GMO space. But even if you want to take a cisgenic or transgenic approach, you can very easily design and build large combinatorial libraries. And the bottleneck is typically the screening of these massive libraries in a reasonable amount of time. With NCAPs, there is some upfront work to design an assay for the desired phenotype and activity. But once that is complete, the mutagenesis and screening is done very, very quickly. So we can typically develop strains with dramatically improved performance, validated in significant time, less than a year. So as Brennan mentioned before, typically in less than 9 months. The other reason why this technology is so special is that it is compatible with a broad range of microbes. So we can really utilize every bug and also very weird bugs if you have very special needs. These include bacteria, yeast, filamentous fungi. We have also used other cell types like mammalian cells or currently also try insect cells. NCAPs has the unique ability to screen these individual cells for a specific phenotype at ultra-high throughput. For example, we can screen nanobody variants for their binding affinity, microbes for their fungal or antibacterial activity, or strains for nitrogen fixation or phosphate solubilization. To give you a little bit a better picture how such an assay and the technology can be applied, I'm going to walk you through 2 case studies that we have done in the past and which should highlight the value of the technology. Angela, the expert here from our AgBio space, will talk you through this prior work afterwards and show how this work can translate to your specific challenge that you have in the agricultural biotechnology. So the first case study I like to show you today is a protease secretion strain. So our partner was producing an enzyme used in the food sector through fermentation with their proprietary fungal strain. To achieve a better cost of goods, they wanted to optimize the strain's enzyme production. And to have a quick path to market for these improved strains, and due to regulatory constraints, they were only considering optimization by classical strain development methods. So our first step was onboard the strain and design an assay that measured improved production through fluorescence, as you can see here on the pictures. Since we always want to directly measure the targeted phenotype, we used a fluorescently labeled protein for determination of protease activity. Degradation of the protein resulted in loss of fluorescence, which can then be used as a metric to determine the level of protease secreted by the strains. From there, we generated a library of nearly 1 million variants from their starting strain using UV mutagenesis. We then encapsulated each variant in a nanoliter reactor with the appropriate growth and assay condition and sorted out the variants that demonstrated the highest level of enzyme production. Among the strains we screened, we isolated the top hits, performed a characterization of the strains with respect to protease production in suspension culture, which confirmed the improved product titer by several strains. At this point, then, we transferred the top candidates to our partner, which confirmed our results in small-scale fermentation. and then selected the 3 best performing strains for pilot scale production, and subsequently the top candidate was selected for industrial scale up and is actually currently now their new production strain. This new production strain had ultimately like a 30% improved titer, which significantly reduced the cost of goods. I think this is really a remarkable result, although 30% seems not too high, but this strain has been previously engineered for decades for high productivity. And within less than a year, we could further improve the strain just to the sole reason that we really had this technology and could analyze 1 million of candidate strains. So Angela, so you have heard the case study. So do you have any ideas for the agricultural space?
Absolutely. Well, first, I think 30% is pretty impressive. And I think that many of our viewers and participants would also think that that's a pretty impressive number, given the amount of time that you talked about and how it had already been adapted. So a few things jump out at me. So many in the environment, and these strains naturally secrete something that protects the plant or provides some benefit to the plant as it's growing. And innovators in this space are very familiar with mutagenesis as a tool to increase the performance of these wild type starting points. It's a fast way to generate a lot of genetic variation without spending a lot of time and money or developing tools to rationally engineer these microbes. So it's a technique that's compatible with the ag space, where innovators are frequently working with weird microbes, and we know you like those, Andy, that are hard to engineer. As you mentioned, once you have this massive library, you're then faced with the problem of how do we test this whole library? There may be only 5 or 10 really high performing strains in a library of 1 million mutants. A needle in the haystack. And as you've shown in this case study, NCAPS technology can answer that question and can do it quickly and, as we'll talk about later, relatively inexpensively. NCAPS enables innovators to turn over every stone and really find that outlier variant strain that outperforms the others in a commercially relevant timeline. And it's a very powerful tool that pairs nicely with mutagenesis. Now back to you. I think you've got another case study.
Yes, I selected one now with another assay principle that I'd like to share with you. So in this case, we were looking for novel antipeptides, which are antimicrobial peptides, as humans and animals face pathogens that are increasingly resistant to traditional antibiotics. So new antimicrobial compounds that we were looking at are desperately needed. So we wanted to test a large peptide library that has been generated in a combinatorial way for its ability to kill pathogenic bacteria. We thought the most direct way to test this would be to co-encapsulate the peptide secreting host cell with a representative pathogen model in a nanoliter reactor, as you can see here in this scheme on the right-hand side. The pathogen model was edited slightly to express a green fluorescent protein so that as the pathogen multiplied, the nanoliter reactor will become more and more fluorescent, okay? Essentially, nanoliter reactors then that were the least green contained the candidate cells secreting a peptide that was most effective in killing the pathogen. Okay, so we ran this co-encapsulation assay and found in the end 126 hits, which form on here on the picture on the right-hand side, the left peak of the graph, as you can see here. So the ones with the lowest fluorescence. The rest of the variants, which formed the right side of this graph, were peptides that were not effective at killing pathogenic bacteria, or in other cases, were not secreted at all. So we isolated the top 126 performing strains and tested them further with respect to peptide secretion levels and also the minimal inhibitory concentration for growth inhibition of a series of pathogens. So these experiments showed that we could efficiently identify novel molecules not existing previously in nature with improved potency against several bacterial strains. So, Angela, I always hear a lot of fungicidal projects flying around. So would not such an assay be also very valuable in this space?
Yes, absolutely, Andy. Totally agree. Crop protection, right, comes to mind. Crop protection chemistry, as we know, is facing all the problems to what you described. Pests are becoming increasingly resistant to chemical crop inputs, and we see a huge opportunity for more biological products to enter the market and address these issues. And we're so excited to enable the innovators who are pushing on these types of products in ag biologicals. And what's cool about this case study is that it's directly applicable to developing future biological fungicides. For an ag partner who is a starting strain with fungicidal activity, we could use this same principle of co-encapsulating variants of their starter strains with a fungal pathogen that acts as a fluorescent biosensor. We could then screen for and isolate the strain variants that have increased potency against that fungal pathogen. And then those strain variants could be generated through mutagenesis, which we talked about earlier. They could also be generated through combinatorial libraries, where we generate a library of amino acid sequences if the active ingredient is a protein. So there are lots of interesting ways we can apply this co-encapsulation assay technique in service of ag biologicals innovation. And more broadly, we can encapsulate cells with other types of biosensors to test for potency. It doesn't have to be whole microbial cells. For other types of pests, insects, nematodes, others, we can find a way to measure potency of the active ingredient in the fabulous nanoliter reactors you described. And instead of validating using MIC assays, we might use some other high throughput assay to validate the hits from the nanoliter reactor screen described. So pretty cool. And we're excited to use this fabulous technology that your team has developed to further ag biologicals. I think we're back to Brennan to wrap it up.
There you go. Put myself off of mute. All right. Thank you very much, Andy. And Angela, I appreciate you going through this case studies because it's important to help our audience understand different ways that we've worked together in this area before and how they would apply to ag. So thank you very much for elucidating that. So for those listening in today, who come away with a better understanding about how this can apply to ag, and I'd like to roughly sketch out what it looks like to work with us. So what makes a candidate really good for this type of a technology? It's when our customer knows what they're looking for in terms of the secreted amount or secreted product. And also if there's an assay that will work together with this ENCAPS technology. And assuming that's the part, that's really what the phases of 1 and 2 are all about. So the first phase is developing the assay that will help us measure the secretion of whatever that target is. Again, a molecule, a protein, an enzyme, whatever it is you need us to secrete more of. And the second part is generating those mutants and finding through this high throughput screening ability, the strain that is now through mutagenesis producing more of the molecule, enzyme, protein at hand. So that's generally what it looks like to work with us from a timeline perspective and sort of what are the requirements in order for this to be a good candidate to work with our ENCAPS technology. There's an optional aspect too about doing a optimization for the fermentation side, which of course, those here on the call probably know is a really great way to boost efficacy as well of the strain. So that's an optional aspect for us to work with this. But all in all, we can do this entire program, including the optional side, and generally under a year. So from that part, then we get to sort of the commercial considerations. And generally what we do for Ginkgo, our philosophy is to share in the risk and reward. So we try to keep our upfront cost as low as possible. And in this case, when we're talking about each strain that we would optimize in this particular process, we're talking about just around $495,000 to do. How that gets paid, we're open to looking at that. And then we're talking about downstream value, which is really where we get to commercialize the product or you commercialize it and see the benefits of working with us. We can figure out what way works best for you, right? Maybe that's a percentage of royalties for running sales, or maybe that sort of lump sum payment that's in consideration of sharing in that downstream risk and reward. And that's really where Ginkgo sees its margin, right? So we try to keep our upfront costs low, give you more access to this really novel technology that, again, delivers dramatically improved strain performance, does so in a really time-efficient manner, and does so without triggering regulatory headaches around the globe, since mutagenesis is broadly enjoyed from a regulatory perspective worldwide. So from a commercial perspective, that's how Ginkgo likes to operate. Certainly, if you have questions, get in touch with us, let us know what some of your concerns are or needs, and we can craft that together. From an intellectual property perspective, we have thought this very through in terms of what needs to be true for you to have what you need to be competitive in the market space with this optimized technology. So broadly speaking, anything that you need to make, use, or sell the product that's patentable will be given to you in an exclusive license going forward. And then sort of the biology that makes this entire thing work, that will be something that would be pertain to Ginkgo. So we can cover that too in a conversation after we have a chance to connect and talk about the commercial considerations and learn more about the strain that you may want to optimize using this technology. So that covers that from that perspective, and just to see if there are any questions and answers. Otherwise, I think we can wrap up for our platform for today. So again, please reach out to us and let us know if you have any questions. My email is listed there below, bduty@ginkgobioworks.com. Would love to hear about how you want to optimize your strains. I think, as Andy mentioned, we can work with a variety of weird bugs. We're excited to do that. And I think there was an answer here too, that we can even work with certain types of plant cells. So let's see if we can solve your problem. And thank you again for your time today. And we look forward to hearing from you soon.